Computational tools today are powerful enough to make a real impact for scientists. This isn't just a dream anymore. With computational tools, scientists can shave years off of development timelines and accelerate breakthroughs. And this matters because time is the one thing that patients don't have. And so we owe it to patients to do everything possible to bring safe, effective drugs to market faster.
Welcome back, smart biotech scientist. You are listening to part 2 of our deep dive into computational bioprocessing with Yossi Quint, who is the CEO and founder of Ark. If you missed part 1, be sure to check it out. Now, we'll explore how in Silico model is revolutionizing tech transfer, decode the science behind virtual bioreactors, and uncover the future of biologics manufacturing. Let's jump right in. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brohlman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. To what extent can we use these models to model the wholesale metabolism and to go into a very granular level to look at all the various parameters and what's going on inside the cell and even predicting, hopefully, quality attributes?
So there's tremendous opportunity for all those things that you mentioned, and especially with quality attributes, which are traditionally very hard to predict. There's been some really great work on mechanistic models. A few academics have done incredible jobs, and we're seeing some of the leading pharma companies also start to do that. But by and large, these are really hard problems to solve, and problems that mechanistic models, for the most part, have insufficiently solved. These types of applications that are on the cutting edge that we've had a really difficult time solving with mechanistic models are the perfect applications for AI.
Now, in the beginning, you mentioned that you're leveraging your experience in finance. To what extent can we leverage models and what's being done in other industries? Can you mention a few examples?
Yeah. So there are a number of, I would say, industry after industry is leveraging simulation for better decision making. And so, as I mentioned earlier, we're seeing that in quant finance, where companies are running thousands of simulations before they make a single trade. But we're also seeing it in the aerospace industry, where instead of having to design an entire jet and test it out and be concerned that it fails after months or years, we could run simulations to understand and to fine-tune every specific part. And so, at the core, across industries, the reason people are using simulation is because they want to cut down on feedback loops. They want to go from feedback loops being driven by physical labor, which is for parts, building, testing, and costly failures, to moving feedback loops in silico, where they can be the timelines can be cut down dramatically. You could iterate many, many more times in the same time period, and you could fail fast and cheaply.
And where should we, as a bioprocessing industry, learn from those experiences? Or where should we perhaps be a bit bigger risk takers or a bit more outside of our comfort zone?
There's always opportunity for us to take more risks. I think it's more about how can we take risks smartly. And in many ways, I actually think that simulation is about de-risking. You could think of it as I'm taking a risk by trying a new technology, but you could also think about it as right now, I am taking a massive, massive risk every time I run an experiment that I have to wait 2 for to get a response. I do believe that in 10 years from now, we're going to look back and say, how could I possibly have tried something new for the first time at a 20,000 liter bioreactor that's costing me hundreds of thousands of dollars when I could have simulated that first and known what the issue was. And so to me, simulation is not about taking a new type of risk, but it's about de-risking.
I love that. This is spot on. It's definitely about de-risking because you can avoid a lot of failure down the road and you understand your processes much better.
I think something that's really cool is that right now there's a bunch of deviations in bioprocess. Things are always changing. Cells are so complex. And it could be because of operational issues. It could be because randomness in the cells or randomness in the material that you're receiving. There's so many different factors, and these factors have outsized impact. And one of the really cool things about simulation is that you could test out hundreds of these common scenarios, of these common deviations, and see how the process responds. And that's impossible today. Can't test out a raw material batch that you don't have. But in simulation, you can. And so by pre-running all these deviations, we could develop much more robust processes. So simulation isn't just about doing what you currently do today better, but it expands the possible.
Yes, it definitely expands the possible. And something also we should talk about is you develop one process and then tomorrow you have another process with a different cell line. I think also that's where the power of your modeling comes into the scale up. Can you elaborate a bit on this?
Yeah. As companies shift cell lines or shift changes of processes, they're going to still want to benefit from the same types of models. And so what we've done at Arc is we've created a modeling approach that is very flexible and that has a bunch of modules or building blocks. And if you think about sort of Legos, like you have a lot of different Lego pieces and they could fit together in different ways, but ultimately they're the same pieces. And so if you have a model that translates the whole size of a sparger into gas exit velocity, into shear, that same model could be used regardless of whether you're doing a cell therapy, a gene therapy, or a MABS. Now, that doesn't mean that you're going to want to use every building block in every single process. If you're running a microcarrier process, you're probably going to want to have a model specific to that that you're not going to need if you're doing a single cell suspension process. But by and large, most of these models you're going to want to reuse time and time again. And so, at least at Arc, we've spent a lot of time building out a very performant code base so that you could turn on and off these models very easily and that the user doesn't have to make those choices, that it's all being done automatically in the back end based on the type of cell line, the type of process that you're using.
And where are the limitations? Assuming we want to transfer some of our learnings, let's say we created our models for a MAPS or a fusion molecule. Can we translate that to a gene therapy or cell therapy process, for instance, or do we still face a lot of limitations there?
So I would say there's two parts to your question, or two ways that I think about it. One of them is, can we use the same modeling structure? And the answer is yes. We're going to need to swap some things in and out, but by and large, we could use the same structure and the same approach. The second part is, can we actually leverage the data from one process that might seem to be dissimilar to a second process? Which, to use your language and the very traditional language of, can we use transfer learning across broader types of molecules? To me, that is the ultimate goal. And if we could use transfer learning effectively, we don't even need the 10 experiments that I mentioned earlier. It could be from day one, we could leverage historical knowledge to make the most of the new molecule. So to me, that is the ultimate goal and the North Star. At least today at Arc, we're not there yet. If you're going within the map space, there's a ton of transfer learning that we could do. But if you're going from a gene therapy to a MABS product, at least today, that's a hard problem to solve.
Yeah, well, definitely the processes are very different. But I would assume that as the models become more powerful and we understand better how to make these models, but eventually we will be able to transfer a lot more learning in the future. Speaking of bioprocess development, what do you think are currently tools or products we are missing to make this whole process faster besides the modeling and less risky? Do you see some other innovations that could facilitate that process?
There are a bunch of innovations that are super high value to scientists. One of them relates to data collection and storage. Right now, when we ask scientists to input data, especially offline samples, into a computer system, or even to take the samples, that's a big burden on them, especially if we want multiple samples per day or if we want samples over the weekend. It's not trivial to ask a scientist to come in every single weekend, every Saturday, every Sunday to take a sample. And because we're not taking as many samples, either the frequency or the duration of those samples, we're not able to build as strong models. And so there's a lot of opportunity for auto sampling and for just reducing the activation energy to do that. Right now in the bioprocess space, we're also still, for the most part, running experiments in a very traditional way. We have a target and we're running the experiment to see if it meets the hypothesis or not. Our view is that if you really want to take a model first approach, a computational bioprocess first approach, you want to rethink the way you're running experiments. So it's not about how can I maximize titer in this DOE or something, but it's about how can I maximize knowledge creation so I could then use a model to find an even better titer, to find a global optima instead of just local optima. And to do that, we need to change the way experiments are run. We want to add more perturbations within the experiment, and we also want to be really thoughtful around when we're sampling. We don't want to just sample maybe at 2 p.m. every day. We want to sample around events and maybe before that event and after the event. The last thing that I'll say is there's so much opportunity for better data capture for new types of probes or for new types of soft sensors. There are tons of ways that we could capture more data in real time. For example, at Ark, we've developed a soft sensor that is able to capture the real time power in a system. Tons of things like this that we could do to have a much more granular understanding of the system and to build much more powerful models.
And now, as we're zooming out, because yes, we want to develop faster, we want to get more process understanding, and eventually we want to get to clinics faster. But we're doing all this because at the end of the day, we are serving patients who need the therapies. So, how does the modeling, whether it's hybrid or another modeling approach, how does this contribute to lower costs? Because we still face a huge accessibility challenge. We have amazing drugs, but they're just too expensive. And how can we leverage all these technologies to enhance patient care and also make sure that more patients get actually access to the therapy?
Yeah, I love that question, David, because ultimately what drives me, what gets me out of bed every morning is that patient question. To me, there's two parts to your question, or there's two ways that we could help patients. The first one, and I would say the most important one, is getting them care faster. If you're a patient waiting for a drug and you have to wait 10 to 15 years for that molecule to come from idea to you, that's just too long. And unfortunately, for many patients, they don't have that time. Time is a luxury that not every patient has. And so the first thing is how can we actually cut down timelines? And that is through running fewer experiments to get to the same answer. And bioprocess is part of that solution, but we also need to bring in faster clinical trials and faster discovery. So it's really an all hands on deck effort. The second part is accessibility, it's price point, and making sure that we're producing enough product that there's enough for anyone who wants it. And that's about delivering an optimized process that has lower costs of goods and a lower R&D cost so that companies can price the product lower and still come out ahead and so that they're not still come out ahead. Today, we spend about $40 billion a year on bioprocess R&D, which is a lot of money and a lot of cost that needs to get recouped. And so if we're able to cut down on experiments 90%, we should be able to spend a fraction of that cost. And if we're able to go from an 18-month process characterization cycle to a 1 or 2 month process characterization cycle, we could cut a year and a half plus off of the process. The last thing that I'll mention is in addition to speeding up timelines and to reducing the cost of goods sold, we can also bring new molecules, more molecules to market. And we started off this conversation by talking about that. If we're able to spend just 10% of the time per molecule in the bioprocess step, it means that we could take 10 times as many shots on goals. The bioprocess teams could test out 10 times as many molecules. And if we're testing out 10 times as many molecules, we're most certainly going to bring more drugs to market. And so a future with computational bioprocess is a future where there are more drugs on market, they're at market faster, and they're at a lower price point so that more patients can afford them. And that's the world that I'm really excited about. Ultimately, will leave the world a much better place.
Yeah, I love that. That's a wonderful vision, Yossi. And I'm just wondering, well, if we do that, if we get a lot more shots and acquiring a lot more data, could also this help us to reduce the attrition rate? Because this is also another problem that costs are so high and it's so risky.
Certainly.
Yeah, that's amazing. Well, obviously, we alluded to that before. We know that we live in a fast changing world. There are new technologies coming up every single day. But let's say, I'm sure you think about that a lot. How do you see your modeling and your activities evolve, let's say, in the next 2 years? That's probably a bit safer than 5 years.
So the first thing is we want to continue to get our models to be more predictive. At the end of the day, what's most important is that the models have incredible predictive power so that scientists can trust them. And that's always our North Star. And so we're always running more experiments in our wet lab, figuring out new ways to increase our algorithms' predictive power. We really care a lot about robust. So that's the first thing. The second thing is expanding into new areas, whether that's new modalities, so many cutting edge modalities on the market, or new parts of the process. Right now, we're really focused on the bioreactor, but we know that there's tremendous opportunity and need on the downstream. Of course, there's going to be new types of technology. We're going to see new advances in modeling approaches. And so we're going to always be on the lookout for those. 2 years is a really long time in this space. And I'm super excited to see what happens in that time frame.
And what is your approach to stay on top of all the innovations?
So things like your podcast is a great way to learn about new cutting edge approaches. Conferences are also a great way. I love going to conferences. I love learning from my peers. And then, of course, there's academic literature and working with universities. In this space, there are universities that are really doing incredible research. And so we have a bunch of advisors who are some of those leading PIs in the world who help us make sure that we're continuing to stay on the cutting edge. And we really owe it to patients to do that.
With all that we've covered today, Yossi, what is your most important takeaway you want our listeners to walk away with?
Computational tools today are powerful enough to make a real impact for scientists. This isn't just a dream anymore. With computational tools, scientists can shave years off of development timelines and accelerate breakthroughs. And this matters because time is the one thing that patients don't have. And so we owe it to patients to do everything possible to bring safe, effective drugs to market faster. I was at a recent conference and one of the speakers said something that really stuck with me, that hit me hard. If patients knew the state of modeling today, they wouldn't be impressed. It's time to change that. We have the technology, we have the data, and now we need to have the will to build something better. And when lives are on the line, there's really no time to waste.
Absolutely. What better way to conclude our conversation, Yossi? Yes, lives are on the line, and this is what drives us. This is what gets us out of the bed every single day, because at the end of the day, it's not just about modeling, even though it's extremely exciting. It's about helping people who desperately need these biotherapies. So thank you so much, Yossi, for sharing your insights, your passion, and it was such a pleasure to have you on. But before we go, where can people get a hold of you?
So I'm very active on LinkedIn, so feel free to drop me a note, send me a message. I love meeting innovators. I love meeting scientists who are on the cutting edge or want to be on the cutting edge, and I'd be thrilled to connect with you.
Hey, biotech scientists, this is your opportunity. Reach out to Yossi. You will find all the links down in the show notes. And Yossi, thank you once again for being on the show today.
Thank you, David. It's a real honor.
And there you have it, smart biotech scientist. The cutting edge of computational bioprocessing from someone who is bringing it to life. If you are ready to optimize your bioprocess development journey, don't forget that I offer free process development assessment. Head over to bromanconsulting.com to book your free call. Until next time, keep innovating and remember, science may give you headaches, but biologics drug development should not. See you in the next episode. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.